Glycoconjugates. A compound containing a carbohydrate component bound covalently to a protein or lipid, forming a glycoprotein or glycolipid. Glycans. Carbohydrate polymers that serve as structural and protective elements in cell walls of bacteria and plants, and in the connective tissue in animals. Carbohydrates. Substances with polyhydroxylaldehydes are ketones, or substances that yield such compounds on hydrolysis. Most have empirical formula of CH2O, N, some may contain N, P, or S. Monosaccharides. Simple sugars, consist of single polyhydroxylaldehyde or ketone unit, most abundant equals D-glucose. Dextrose, simple sugars with 4 or more units tend to have cyclic structure. Oligosaccharides. Consist of short chains of monosaccharide units, residues, joined by characteristic glycosidic bonds. In cells, most of these are not free, but rather bound to non-sugar molecules in disaccharides. Most abundant form of oligosaccharides with 2 monosaccharide units. Example equals sucrose, cane sugar, D-glucose and D-fructose units. Polysaccharides. Sugar polymers containing more than 20 or so monosaccharide units. Examples equals cellulose, linear chain, glycogen, branch chain. Both of these examples consist of recurring units of D-glucose, but they differ in the type of glycosidic linkage, and thus have very different biological roles. Aldose. Simple monosaccharide with the carbonyl group at the end of the carbon chain, forming an aldehyde group. Simplest example, glyceraldehyde and aldotriose. Ketose. Simple monosaccharide with the carbonyl group at position other than the end of the carbon chain, forming a ketone group. Simplest example, dihydroxyacetone, acetotriose. Tetrose. Monosaccharide with 4 carbon atoms in the backbone. Pentose. Monosaccharide with 5 carbon atoms in the backbone. Nucleic acids and nucleotides are made of aldopentoses, D-ribose and 2-deoxy-D-ribose. Hexosa. Monosaccharide with 6 carbon atoms in the backbone. Most commonly found monosaccharides in nature. Aldehexoside-glucose and catahexoside-fructose. Heptose. Monosaccharide with 7 carbon atoms in the backbone. Chiral character of monosaccharides. All monosaccharides, except dihydroxyacetone, contain 1 or more asymmetric carbons and thus occur in optically active isomeric forms. Enantiomers. 2 different optical isomers of a molecule. Example, glyceraldehyde, simplest aldose, containing 1 chiral center on the middle carbon. X-ray crystallography. used to determine the absolute configurations of sugars and other biomolecules. Fischer projection formulas, used to represent 3D structure on paper. Horizontal bonds project out of the plane towards the reader, and vertical bonds project behind the plane, away from reader. Molecule with N-chiral centers has 2 N stereoisomers, D-isomers. Those having the same configuration around the chiral center farthest away from the carbonyl carbon, as D-glyceraldehyde. L-isomers. Those having the same configuration around the chiral center farthest away from the carbonyl carbon, as L-glyceraldehyde. Epimers. 2 sugars that differ only around 1 carbon atom. Examples of sugars that occur in L form naturally: L-arabinose and the L components of some sugar components that are part of glycoconjugates. Cyclic ring structure. In aqueous solution, all the tetroses and all 5 or more carbon chains in backbone structure occur, predominantly in cyclic ring structure, in which the carbonyl group covalently bonds with the oxygen of a hydroxyl group along the chain. Hemiacetyls, hemicatyls. Derivatives of a generalized reaction between alcohols and aldehydes, ketones. These contain an additional asymmetric carbon atom and thus can exist in 2 stereoisomeric forms. Pyranoses. 6-membered ring compounds formed by cyclizing the straight chains, named this because they resemble the structure of pyran. Furanoses. 5-membered rings made by cyclizing straight chains, so named because they resemble furan. Stability of pyranoses versus furanoses. 6-membered aldopyranose ring is much more stable than the aldofuranose ring, and predominates in aldehexose and aldopentose solutions. Anomers. 2 stereoisomers of a given sugar that differ only in the configuration about the carbonyl carbon atom. Anomeric carbon. The carbon atom in a sugar at the new stereocenter formed when a sugar cyclizes to form a hemiacetal. This is the carbonyl carbon of aldehydes and ketones. Mutarotation. The process of interconversion of alpha and beta anomers in aqueous solution, mentioned in D-glucose. Haworth perspective formulas. A method used for representing cyclic chemical structures so as to define the configuration of each substituent group, commonly used for representing sugar. What is the general formula for a carbohydrate? CNN. List the characteristics of a monosaccharide. Hurry. The aldehydes are ketones with two or more hydroxyl groups. Aldoses contain an aldehyde group. Retoses contain a ketone group. What is the difference between an aldehyde and a ketone? An aldehyde has a carbonyl group at the end. A ketone does not have its carbonyl group at the end. How many carbons are in a triose, in a heptose? There are 3 carbons in the backbone of a triose. There are 7 carbons in the backbone of a heptose. If a molecule has 5 chiral centers, how many stereoisomers can it have? 2 stereoisomers, and chiral centers equals 2 stereoisomers. What is the difference between L and D monosaccharides, which is more common? The L isomer of a monosaccharide will have the hydroxyl on the chiral center furthest from the carbonyl group on the left side. The D isomer will have the same hydroxy on the right side. Unlike proteins, which are L isomers most common, monosaccharides have D isomers as the most. Define epimer. Epimers are 2 sugars that differ only in the configuration around one carbon atom. What happens to monosaccharides in aqueous solution? What is the minimum number of carbons necessary to do this for a doses? What groups are involved in forming this bond? In aqueous solution, monosaccharides occur predominantly in cyclic structure, need minimum of 5 carbons. The groups involved are a carbonyl group to form a covalent bond with the oxygen of a hydroxyl group in the chain. What is an anomer? An asymmetric carbon formed with the formation of a cyclic structure that can have either an alpha or beta configuration. Alpha is hydroxyl on different side of CH2O moiety. Beta is if it's on same side. What is a mutarotation? The conversion between the alpha and beta configuration. Define electrophilic. The cellophilic. When forming a ring, which group is the nucleophile? Electrophile, electron-deficient group with strong tendency to accept electrons. Nucleophile, an electron-rich group with strong tendency to deny electrons. When forming a ring, the alcohol acts as the nucleophile. Define hemiacetyls and hemicatyls. Hemiacetyls form when aldehydes are attacked by alcohols, have one R group and a hydrogen. Hemicatyls form when ketones are attacked by alcohols, have 2 R groups. What is the difference between a pyranose and a furanose? A pyranose is a 6-membered ring containing oxygen. A furanose is a 5-membered ring containing oxygen. Briefly describe 3 classes of important sugar derivatives. Amino sugars. NH2 replaces O. Some amino acids often condensed with acetic acid. Deoxy sugars. H replaces O*. Look at book. Acidic sugars. C atom is oxidized to carboxyl group, phosphate group. Addition traps sugar in cell, activates it for chemical transformation. Define reducing end, reducing sugar, and non-reducing sugar. Reducing end, the end of the chain with a free anomeric carbon that can be oxidized, reducing sugar, the reducing end that refers to the anomeric carbon not in a glycosaminoglycans, non-reducing sugar. Explain the abbreviation system for D. Polysaccharides. What are abbreviations for GLC and GAL? Drop the D from the name. Show that old anomeric carbon was in the Xelve form. Show which carbons. The linkages between GLC get help Define and give examples of homopolysaccharides and heteropolysaccharides. Homopolysaccharides, single monomers used for energy storage, starch and glycogen. And structural components, cellulose and chitin. Heteropolysaccharides, 2 or more monomers used for extracellular support, and extracellular space, which is the matrix that gives protection, holds cells together, and creates shape and support. Which homopolysaccharides are used for the storage of energy? Are they hydrated? Why? Starch and glycogen are used for storage of energy. Starch plants and glycogen animals. They are heavily hydrated so the O groups can form H bonds with water. What are the 2 components of starch? Are they branched? What types of linkages are in these molecules? Starch mixture of 2 homopolysaccharides of glucosa. 1. Amylose, long unbranched chains, linear. Polymer of D-glucose with alpha 1 to 4 linkages. 2. Amylopectin, branched, has alpha 1 to 4 linkages. Like amylose but branches with alpha 1 to 6 links, branches every 24 to 30 residues. In what form is starch stored? From which end are residues cleaved to mobilize energy stores? Starch is stored in granules. Residues cleaved enzymatically from the reducing ends of outer branches during mobilization of starch for energy production. If glycogen has 6 branches, how many non-reducing ends does it have? Glycogen with N branches has N1 non-reducing ends, 7 NREs. Describe two homopolysaccharides used for structure. Where are they found? What monomers are they composed of? What type of linkages do they have? One. Cellulose found in plant cell walls, linear, unbranched, 10,000–15,000 glucose residues, have B1–4 linkages, similar to amylose except in linkage, hydrogen bonding occurs between linages. 2. Chitin found in cell walls in mushrooms, and in exoskeletons of insects, linear. Homopolysaccharide of N-acetylglucosamine has B1–4 linked chain. In polysaccharides, where does the flexibility of the molecule occur? The flexibility lies in the bonds in between the monomers, because monosaccharide rings are rigid. Which polysaccharides bonds can freely rotate? What are fine psi? CO bonds that link the monomers can freely rotate dihedral bonds of a glycosidic linkage phi and psi, but some steric hindrance from atoms on the ring does affect rotation. What types of bonds stabilize the 3D structures of polysaccharides? Stabilized by H bonds, many are groups, hydrophobic interactions, van der Waals interactions, electrostatic interactions. If they contain substituents, think sugar derivatives. What 3D structures are found in starch and glycogen? Which is most regular and why? How does cellulose differ? Tightly coiled helix structure. The most regular is a helix in amylose, which doesn't have any branching. Cellulose stretches out in a linear fashion, rotated about 180 degrees compared to neighbor. Cellulose hydrogen bonds form between no groups of adjacent molecules to give rigidity. Cellulose needs Briefly describe the ECM. What functions does it perform? Gel-like extracellular space in tissues of multicellular animals, holds cells together, provides support and anchorage for cells, helps regulate intercellular communication, contains many proteins, structural and functional. What are the major heteropolysaccharides found in the ECM? Glycosaminoglycans. Describe the characteristics of glycosaminoglycans. Linear polymers of repeating disaccharide units. Negative charge attracts water molecules and keeps ECM and cell hydrated. Repulsion of negative charge keeps molecule extended with rod-like helix, unique to animals and bacteria, not plants. What are the disaccharide units making up GAGs usually made up of? One of the two is always N-acetylglucosamine or N-acetylgalactosamine, the other is usually a type of uronic acid. Carboxylate group has negative charge. List 4 GAGs, which are sulfated. Which ones contain uronic acid? greater than hyaluronic acid, only one without sulfates, and is the largest, greater than chondroitin sulfate, greater than keratin sulfate, only one without uronic acid, greater than heparin sulfate, has the most sulfates. What is the function of the negative charge on these molecules? What is a glycoconjugate and what are some of its functions? Lipids are proteins covalently attached to a carbohydrate, functions to carry information for cell-to-cell and cell environment communication, for recognition sites for extracellular signaling, molecule, for cell migration, to label proteins for transport. What are the three main types of glycoconjugates? Proteoglycans, glycoprotein, glycolipids. Define glycocalyx. An outer carbohydrate layer several nanometers thick. What is a proteoglycan and how is it attached to a core protein? Sulfated glycosaminoglycan chain joins to membrane proteins located on the surface of the cell. Briefly describe types of protein interactions of proteoglycans that mediate functions. Describe proteoglycan aggregates. Enormous supramolecular structures in the ECM, 20 million Mr. Many core proteins associated non-covalently to a single molecule of hyaluronan. Core protein called aggregan must have a lot of consensus sequences attached to the core protein equals GAGs through saccharide bridges, interacts very strongly with collagen and helps with the strength of connective tissue. What do proteoglycans interact with and what effect does this have on the ECM? Interact with collagen and other molecules like proteins, fibronectin, fibrin, integrins, other GAGs. Interactions form a meshwork that gives ECM strength, anchors cells, conveys information in both directions, helps ECM with major functions. Briefly describe glycoproteins. What is the difference between unlinked and dolinked? A protein with small oligosaccharides covalently attached can be one or more carbohydrate. Attached via anomeric carbon, O-linked equals glycosidic linkages to the O group of seride are called O-linked glycoproteins, and linked equals glycosidic linkages to the amide N of ASN. Briefly describe glycolipids. Lipids with covalently bound oligosaccharide, usually membrane-bound lipids, are attached to oligosaccharides. Phospholipids contain hydrophobic fatty acid tails and have a hydrophilic head group containing phosphate oligosaccharide attached to polar head of the lipid. What is meant by sugar code? Refers to the amount of information a sugar can hold that is used for cell-cell interactions, signaling, cell adhesion, and targeting protein to specific places. Why can carbohydrates hold more information than proteins? Carbohydrates each have a number of monomers which can then be altered by addition of more monosaccharides, which can then each be altered with groups added, and can each have different binding patterns equals billions of combinations. These codes are read by the different protein they interact with. All of these components will code for different functions of the cell, so they can store massive amounts of information. What is a lectin? Describe 2 examples of lectins in action. Lectins are a group of proteins that read the sugar code by specifically interacting with specific sugar, recognizes oligosaccharides in glycoproteins and glycolipids. 1. White blood cell movement. To a site of infection or injury, neutrophils slowed and moved to site of inflammation by interactions with integrins and p-selectins. Lectins. 2. Virus host and bacteria host interactions. The flu lectin protein binds to ligand on virus cell surface. Define multivalent. What does this term have to do with lectins? A lectin molecule has multiple carbohydrate binding domains, can attach to a number of specific sugars which can increase its affinity. What is the difference between avidity and affinity? Affinity is how attracted a ligand and lectin are to each other. Avidity is effective affinity, and is increased by the multivalency of lectins. Get help. Define KD. Equals dissociation constant given in molar concentration, a measure of the affinity for a protein to its ligand, equivalent to the molar concentration of ligand at which half the available ligand and binding sites are occupied. Does pH affect binding interactions between lectin proteins and sugars? Yes, if pH is lowered or higher, it could change the charge on the amino acid residues of the protein and alter the environment of the binding pocket. Change could cause release of sugar. What types of bonds occur during a lectin-carbohydrate interaction? Multiple weak bonds involved, H-bonds and other hydrophobic interactions. What is meant by the term glycomics? The characterization of all carbohydrates in a given tissue or cell includes mono, D, polysaccharide and all glycoconjugates and where they are attached to proteins or lipids.